Well, good evening, everybody, and welcome to this evening's lecture. I'm John King, founder of the Shrewsbury Darwin Festival, and it's my pleasure to introduce this evening's speaker. Before we start, a quick word on the format for today. This session is being recorded for our archives. At the bottom of your screen is a Q&A button, and you can use that to post any questions you have. And please state your name and address, your name and where you're joining us from. That would help too. At the end of the lecture, if we have any questions, I'll be putting some of those to our speaker in a short Q&A session. So without further ado, our speaker this evening is Dr. Simon Jeffery. Simon is a soil microbial ecologist at Harper University here in Shropshire. The title of this evening's lecture is Life Beneath Your Feet. We're living to some extent in a virtual world at the moment. Lockdown has separated us from our family and friends, and here we are hosting a festival on laptops. But for too long, we've been separated from the realities of how the things that sustain us are grown. In the lecture this evening, Simon will address the subject of soil degradation and how we ignore it at our peril. Simon, over to you.
Okay, hi John, hi everybody. Thanks very much for that introduction, and thanks a lot for the possibility of doing this presentation today. If you just bear with me while I sort the technology out. Right, hopefully what you're all now seeing is a PowerPoint presentation just appearing on your screens. So yeah, my name is Simon Jeffery. By way of introduction, I'm a reader in soil ecology at Harper University, and I'm going to spend some time this evening talking to you about a topic that I've been researching for about the last 15 years, and that's the life beneath our feet. Now, it can be surprising to some people that you could spend so much time researching the life below ground, and that's because we normally interact with soils in this kind of way. We see soil as this two-dimensional flat surface that stretches off towards the horizon. It's generally quite unremarkable, and it's a non-exciting brown colour, so it's quite hard to capture people's imagination with it. Even when there are hills, when there's relief, which obviously requires that third dimension for that relief to fit in, we still only generally interact with this soils as this two-dimensional flat surface. So that belies the fact that below ground there's actually this highly complex environment that's teeming with life. In fact, in both of these pictures, there is more life, there's more biomass below ground than you can see above ground in these places. On average, UK grassland contain the equivalent of about 2,000 sheep per hectare in terms of the amount of weight of all the living stuff that you find below ground. Now, I suspect that sheep per hectare isn't a unit that many of you are used to working with. I suspect many of you are more used to dealing with things such as blue whales or double-decker buses. So to provide a bit of context for that, the maximum stocking rate for sheep within the UK is 12 sheep per hectare. So you can see there's orders of magnitude more life below ground than we find above it. Now, as a soil scientist, a soil biologist, we actually spend some time digging down into soils, and we see things like soil profiles that you can see in front of you. And so obviously to do this requires depth. We're now going down into that soil environment. And whilst digging these pits, you might see the occasional worm or an occasional beetle scurrying around, but you certainly don't see 2,000 sheep's worth of life down there, which leads to the obvious question, well, where is it? Where is the life? Well, the reason we can't see it, of course, is because it's all about scale. The type of scale that we normally interact with soil in, depending on how tall you are, generally somewhere between 1 metre or 2 metres above the soil surface. We walk around as these kind of eyes suspended above the soil surface, and we know that there's this depth of soil underneath us, but it feels like this solid mass because we don't sink down into it. You can drive tractors over the top of it and they don't sink down into it terribly far. So it's hard to imagine that there's so much space for life down there. Now, if any of you are gardeners, if any of you have dug down into soil, you might have seen soil at this type of scale. You can see the scale bar at the bottom. And at this scale, we can start to see there's some texture, there's some gaps between the soil particles, there's some organic materials mixed within the soil. However, to appreciate the space that exists within the soil, we really need to look down a microscope. So this is using a stereo microscope, and you can see here some plant roots growing down through the soil and some mycorrhizal fungi coming off those, which we'll get to in a minute. But what you can start to notice at the top and bottom of this slide is there's space there, there's gaps within the soil. If we use a compound microscope, we can get down and view the level of microorganisms. And at this scale, this is a thin section that's been taken through the soil. All of that yellow, that's light that's shining up through this thin section of soil from the bottom. That's the pore space. That's where life finds its home. Now, if you could take an average soil, if such a thing could be said to exist, and take a cubic meter of that soil and lift it up and divide it out into all its parts, about 50% of that cubic meter would be pore space. There's a lot of space once you get down to the microbial scale, and that's how we can fit so much life into these soils. Now, I keep saying microorganisms and small spaces, and I just showed you an image with a scale bar, but it's still quite hard to visualize just how small micro is. So in order to try and help visualize that a little bit, if you were to blow up a millimeter to the size of this bar, well, a human hair would come in at about that type of diameter, about 50 micrometers across. An amoeba, which we'll see one of in a minute, they exist at about that sort of size. They're about the same diameter as a human hair. They're really right about at the limit of what a human can see with a naked eye. If you have one in the bottom of a petri dish and you've got good eyesight, you can just about make out there's something in there. And if you watch it for long enough, you'll see that it's moving around, but you can't make out any of the cellular structures. Now, the other microorganisms are actually generally much smaller than that. So we have a nematode worm, again, we'll have a look at some of those in a minute, that image on the right, and some fungal hyphae. And then in that smaller image, you can hopefully see clustered together some bacterial cells. Now, those bacterial cells are a realistic size for bacterial cells that you would find within the soil, but they're actually about as big as bacterial cells would naturally be within the soil that are commonly found. The reason why I've chosen the biggest ones to include there is because if I chose the average size ones, you basically wouldn't be able to see them at this scale still. To give you an indication of just how small they are, hopefully you can all see that square between the 10 micrometers and the picture of the bacterial cells. Well, average size bacterial cells, you could fit 100 of them on that little square there, just to give you an indication of the sorts of scales that these organisms exist at. Now, we've been interacting with soil so far as this two-dimensional flat surface, and as I said at the start, that belies the fact that there's this complex three-dimensional environment below ground. So I'm going to show you a short video which hopefully helps to highlight just what the environment below ground is like. Now, this is going to look like computer graphics, and indeed it is computer graphics, but it's certainly not a computer game. What this is, if I start the video, this is a real soil sample. A cubic millimeter has been very carefully sampled and placed in an X-ray tomograph instrument. And that basically takes lots of X-rays through this images and spins it around and takes more X-rays, and then you use a computer to reconstruct the internal structure of that soil aggregate. So if you could squeeze yourself down to the size of an amoeba, this is how the soil environment looks to you. It's this labyrinth of three-dimensional pores of very high complexity. Some of them will be filled with water, some of them will be filled with air, lots of organisms moving around. And it's because of this complex environment of all these small pores, that's how we can fit so much life into soil. Now, it can be quite hard to imagine what an amoeba squeezing through pore space looks like, but fortunately, we don't actually need to imagine it. This is an image which was taken some years ago by Carl Ritz, who's actually my PhD supervisor. And what you can see here is an amoeba actually squeezing through soil pore spaces. They don't normally glow like this. This is a type of microscopy called epifluorescent microscopy. And these organisms go around through the soil grazing on bacteria that they can find there. So, which organisms live within the soil? I'm going to take you as a bit of a whistle-stop tour through some of the major groups. I can only talk for 30 to 40 minutes to allow time for questions, so I'm going to try and keep waffle to a minimum, but I'm going to introduce you to the main groups. So we normally split organisms up that live within the soil into different size categories. The megafauna, that's things like badgers, foxes. I'm not going to talk about them at all today because they don't really do any soil functions that are important for soil health. The biggest organisms we're going to look at are organisms that I suspect you're all aware of. They're the earthworms. But within the macrofauna, we also have ants, woodlice, centipedes, and so on. These are organisms that, by definition, their adult forms are above 2 millimeters in size. Coming down in size, from 100 micrometers to 2 millimeters in size, we have the mesofauna. 100 micrometers is 1/10 of a millimeter, just to help with the scales. At this size, we have tardigrades, the common name for which is the water bears. We're not going to look at them today because we don't really have time. The main groups we're going to be looking at are collembola and mites from this group. Now, the mesofauna, you can generally see these with the naked eye, but you can't make out too much detail. You can just see little organisms scurrying around. Now, if you could take a big handful of an average soil and you could extract all of these organisms, you would expect to find thousands of individuals from hundreds of different species just in that handful of soil. Coming down in size then to the microfauna and flora, these range in size from 1 micrometer up to 100 micrometer, and this consists of the bacteria, the fungi, and the protozoa. An amoeba is a protozoa that we saw just now, and those nematode worms. Now these can exist in very remarkable levels of abundance within soils. If you take a good handful of soil, you would expect to find more bacterial cells in that handful of soil than there are human beings on all of planet Earth combined. If you had the patience of a saint and a good pair of tweezers and a microscope, you could go through and pull out all these fungal hyphae, which are these fine filaments. They look like hairs growing through the soil. Again, in that same handful of soil, you could expect to find 50 kilometers of hyphae. And it's the fact that we have such large abundances, such large amounts of these organisms, that despite their small size, they really do perform functions that are vital for the maintenance of life on Earth, as we will see. So all of these organisms interact with each other in ways that I suspect you're all aware of, because it's the way that organisms above ground interact. And that is basically things run around trying to eat and trying to avoid being eaten. Now, the way above ground things work is the light shines down on green things, on plants, and they photosynthesize. They basically take energy from the sun and turn it into plant material. They lock up some of that energy. And that energy then gets passed up the food chain through primary consumers and so on. Of course, the real world is much more complicated than this. Most things don't just eat one thing, and they're not just predated by one predator. There's a whole food web that occurs in a natural environment rather than these food chains. And below ground is very similar. It's just everything occurs on a much smaller scale. Another difference is within the below ground environment, we also have a different pathway. We have kind of a secondary pathway where materials can be recycled. And that's the organic matter pathway that we can see down here. The organic matter pathway, that's materials such as leaf litter. In autumn, many leaves in this country, of course, fall to the ground and they get decomposed. Well, that decomposition is feeding the organisms of the soil. Also manures, dead animals, all of these other materials, they make up the organic matter and they get decomposed over time within the soil. And that decomposition is very important because there's lots of nutrients within organic matter. There's lots of nitrogen, phosphorus, potassium, all of these things that plants need to grow. And they can't access any of those nutrients when they're tied up within organic matter. We need biology to go in there and to burn off the carbon to liberate the nutrients that are there. Technically, we call it mineralizing, but basically they free these nutrients, which then plants can take up and they can carry on being cycled through the ecosystem. If we don't have this decomposition pathway functioning properly, or we don't have enough organic material to feed the system, then ecosystems can become much less resilient. They can start to become a lot more susceptible to not functioning so well under stresses such as climate change. Right, so I'm going to start with the largest organisms we're going to look at today, which is the earthworms, and give you a very brief introduction, and then we're going to move down through size categories and finish with some bacteria. So here we have some earthworms. I'm sure you're all aware of what earthworms look like. I suspect many of you have come across them on a fairly regular basis. Now, earthworms, within the UK, we have 27, arguably 28 species of earthworm, and they can be divided up into what are generally referred to as ecotypes. They all basically live in slightly different ways or interact with the soil in different ways. First, we have the epigeic earthworms. These are the ones that you can see at the top. These live generally on the soil surface, and they eat leaf litter and other detritus on the soil surface. We have the endogeic earthworms, and the second ones that you can see. They generally burrow horizontally within the soil surface. They very rarely come up to the surface themselves, and they leave all of their casts within the soil as well. So you don't see earthworm casts from these organisms very often. And then finally, we have the anisic earthworms. These are generally the most famous earthworms. They form these long, permanent, semi-permanent burrows down into the soil. Some of them can go down as much as 1 meter deep, depending on the soil type they're in. And they come out at night time and forage around on the soil surface. They put their head out and pull organic materials down into the soil. And they produce their casts on the soil surface. So if you've seen earthworm casts, it will generally be from this ecotype. Now, all of these earthworms together, they function to turn over the soil and incorporate organic materials where they can be decomposed in environments that they make nutrients available for plant uptake. So they're very important for plant productivity. And because plant productivity is the basis of above ground ecosystems, they're very important for ecosystem health. Now, this has been known for a very long time. Darwin himself, in fact, published a book back in 1881 called The Formation of Vegetable Mold Through the Actions of Worms, with observations on their habits. And actually, this book sold 6,000 copies in its first year, which is more than On the Origin of Species managed in its first year, which just goes to show it wasn't only Darwin back then that understood the importance of worms. It was a well-known phenomenon that if you have more earthworms in your soils are more productive, you get better yields. Now, science has come on quite a long way since the time of Charles Darwin, and there's been a lot of experiments done. And one that was published back in 2014 looked at the effects of earthworms on crop yields. Now, this was a particular type of study called a meta-analysis. So this type of study, this research, wasn't done by going out and doing an experiment. Rather, these researchers looked at all of the published studies that they could find where people had done experiments where they compared plant growth, crop productivity, when earthworms are present compared to when they're not present. And what you do for meta-analysis is you take the data from all of those studies and you build a database, which is very time consuming and a bit of a pain in the neck. But once you've done that, you can then apply some statistics to that data, which is a bit beyond the scope of anything we can discuss here, although feel free to contact me via email if you want to know more. But once you interrogate the database with statistics, you can produce figures like this. Now, the statistics themselves are quite complicated, but understanding these figures is thankfully very simple. So what we have here is the earthworm effect on the x-axis, okay? This y-axis is just here to show the 0 mark. You don't need to read anything against that. So the way to interpret this is this point shows the mean effect. So in terms of crop yield, the average effect of having earthworms present compared to not having them present is somewhere around a 25% increase in crop yields. Now, as a farmer, if someone tells you, well, if you can manage your soils well to increase earthworms, that can increase your yields by 25%, well, that's quite a big incentive because 25% increases in yields are not easy to come by. Now, for the more statistically minded in the audience, just to finish this up, these bars represent something known as the 95% confidence intervals. For the non-statistically minded in the room, you don't need to worry about that. That's just a measure of how much variance there is in this data. So we think the mean is somewhere around 25%, but it could fluctuate somewhere between about 15 and 35%. Most importantly, these error bars don't intersect the 0 line, so we're confident in this result. It's what we would call in science a statistically significant result. So the earthworm effect statistically increases yields, above ground biomass, below ground biomass, and total biomass. All of the three, the main aspects that are required in order for you to have ecosystem health. However, earthworms don't affect the ratio of shoot to roots. They seem to increase both above ground and below ground biomass, as shown by the figures above. And they don't increase the amount of nitrogen that's available in the plants in terms of nitrogen concentration. So this recognition of the importance by Darwin back in 1881 has certainly since been validated very well since then. Now that's it for earthworms, and I'm afraid I don't really have time to talk about many of the other soil macrofauna. Here's an image of some of them. They include things such as carabid beetles and pseudoscorpions. The main role for these is some of them can be very important predators of pest organisms such as aphids, and some of them can be also important for shredding and breaking up plant litter to help speed up the decomposition processes. But that's it for the macrofauna. We're going to jump down in scale and have a look at some of the mesofauna. To start with, we're going to have a look at some collembola, the common name for which is springtails. Now, within the UK, we have somewhere between 250 and 300 species of collembola, and these generally exist around about a millimetre, sub-millimetre in size. Now, it's often said by people who work within soil research that it's quite hard to engage the public with soils because we lack charismatic organisms. If you talk about conservation, then most people think about tigers and pandas and other very charismatic organisms. And when you talk about soil, most people think about things like earthworms, which, as important as they are admittedly not terribly charismatic to most people. However, to say there are no charismatic organisms in a soil generally suggests that people who think that have not seen enough collembola and certainly haven't seen enough globular collembola, such as this little chappy that you can see in the middle at the top of this slide. So these collembola can also be divided up into ecotypes, into different categories depending on how they live, in the same way that we can with earthworms. And that can be done very easily just by looking at their morphologies, at their characteristics and their shapes. So the first ones, as you can see on the top left, we have the epigeic. That just means surface dwelling. These organisms generally have large antennae. They're normally pigmented because, well, they need to be camouflaged so that things can't find them to eat them, and they need protection from the sun, from the UV of the sun. They also generally have springtails. They have these things called furcula, which you can see in this bottom image down here. This is the springtail. That's normally bent around underneath their belly, and when they feel threatened, they flick that tail down and go jumping up in the air. Now, these organisms have sometimes been claimed to be agricultural pests, but they're certainly not agricultural pests. They play a role as detritivores. Basically, they shred and break up organic materials within the soil. And actually, some of them can play the same role as pollinators, such as bees play with flowers. They do the same with mosses within the soil. So none of them that I'm aware of, at least, are pests. So we have also, next group is these hemiadaphic, these two that you can see on the right. These are generally smaller. They have smaller appendages and smaller antennae. Some of them don't have eyes or ocelli, as they're technically called. And these organisms move between the surface and the subsurface environment. And then over on the bottom left, we have the euadaphic. They're the true soil-dwelling organisms. They generally don't come to the surface, so they're not exposed to UV, and they're not visible to other organisms, so they don't need any pigmentation. They live within a dark world below ground, so they don't need any eyes because they can't see anything anyway. And they don't have a springtail, because if you're underground, there's not much point in flicking a springtail down because you don't escape your predator. You just get eaten with a headache because you've just banged your head against the soil pore above you. So these are the Collembola. Very quick introduction to them. Moving on to have a look at the mites, the technical name for which is the Acari. These are an incredibly diverse group of organisms. On planet Earth, it's estimated that there's probably more than 1 million species, and many people who are Acari experts actually claim that there's more diversity within this group than there are within the beetles. Now, many beetle experts dispute that, and there's a bit of an ongoing spat between them, which I generally stay out of. But nevertheless, they are certainly a highly diverse group. Here are some more images of them just to show some of the different morphologies. Now, again, the vast majority of these are detritivores. The main role that they play within the soil is chewing up the organic material and speeding up the decomposition rate and facilitating and helping that decomposition, which frees up nutrients for the wider ecosystem. However, some of these organisms can actually be important agricultural pests. You can see an image on the left here. That's damage that's been caused by a two-spotted mite, which you can see a scanning electron micrograph there of on the right. So some of these can be important plant pests, but they also present a means of controlling those pests, because some of these mites are predatory organisms. And this predatory mite that you can see on the left there is a predator of the two-spotted mite. So there's quite a lot of research going on, and in fact at Harper we have a group that's doing research with this group of organisms where these can be bred up, and generally they're not released into arable or outdoor ecosystems, but within horticultural systems, within greenhouses and these sorts of productive ecosystems. It's been shown that they can actually control these pests very, very effectively. And using these means you don't have to use any other chemistry, any other nasty pesticides to try and control those pests. And these organisms are quite species specific, so they only attack these pests that they like to eat. They leave other beneficial organisms around, which of course, unfortunately, most pesticides don't. I can't really talk about mites or Acari without mentioning these two. We have perhaps the most famous mite over on the left, which is the deer tick, and that's famous not for good reasons. That carries Lyme disease, and in some areas of the country, Lyme disease can be highly prevalent within their populations, unfortunately. The one on the right, the Lone Star tick, perhaps from the name you can well imagine that that comes from America, from Texas, where it was first found. The closest that one has been found to the UK was at Belfast Airport, where one was found on someone's clothing, but fortunately it was captured and killed before it got released into the environment. And that's a good thing, because we don't really want these over in the country. If you get bitten by one of these, it can cause something called alpha-gal allergy, which I suspect you may not have heard of. The consequence of alpha-gal allergy is it basically causes your body to have an immune response to a protein that is found in mammalian blood. Now that in itself might not sound too bad, but the consequence of that is basically it makes you allergic to eating red meat. So if you are a carnivorous person who enjoys eating meat, or if you are a livestock farmer, it's certainly not the sort of organism we want too many of within this country. or any country, to be fair. Right, the mesofauna, again, they're another very diverse group, including organisms that can be very small. You can see a pseudoscorpion down at the bottom. We have bristletails and enchytraeids. Most of these organisms, the roles that they play, again, is speeding up this decomposition process and cycling nutrients through the ecosystem. Some of them can also be important in terms of pest control as well. But as time is limited, we're going to move on from the soil mesofauna and go down into the microfauna. Before that, though, I almost forgot about this slide. These organisms have been used quite a lot in research as indicators of soil disturbance. So here we have an image published by Christina Menter back in 2012. And basically the way that it works is you look at these organisms and you look at the columbella and the acari, and you look at the characteristics that I've mentioned, whether they look like they're surface dwelling, so they're pigmented with long limbs and long antennae, or whether they are subsurface dwelling, so they're not pigmented and they have short limbs and they don't have furcular and all of these things. And by looking at that and these different characteristics, you can give them a score. And these scores have been shown to correlate very readily with how disturbed a system is. So you can see over here, we have sugar beet on the left. That's just about as disturbed as a soil environment can get, because sugar beet are a root crop, so you have to disturb the soil a lot when you're planting them and when you're harvesting them. At the other end of the scale, we have wood. We have forested areas where soils are very undisturbed and they generally have large organic matter contents because of that leaf litter going in. So by looking at these organisms and by applying this scale, we can actually start to try to quantify soil health, or the level of disturbance that we think exists within particular environments. And we can start to look at how soil management practices and changes in those soil management practices affects these soil communities. Right, now then, coming down into the microfauna and flora, here we have a nematode worm. Again, this is epifluorescent microscopy. They don't normally glow blue like this. They're actually transparent when you look at them down a microscope. Now, for these organisms, from this size down, they are basically too small to be able to move soil particles around, so they're completely constrained by the soil environment. Some of these can be important plant pests, things like potato cyst nematodes, or here we have root knot nematodes, can really cause quite a lot of agricultural damage and a lot of financial damage to farmers. However, these also represent a means of controlling those pests. There's a type of nematode called entomopathogenic nematodes, which attack these insects and are capable of knocking back pest populations. And much like the predatory mite I mentioned just now, these are highly species specific. So if you choose the right nematode species and release it into an environment, it will only attack the pest that you are after attacking. So there's a lot of potential going on. And again, there's lots of research going on into these organisms at Harper University. So on to the other microorganisms that are perhaps a bit more well known. We're going to have a look at the fungi. So here we have some images of the above ground parts of the fungi, but actually these organisms exist with the vast majority of their biomass below ground. And here you can see these filamentous hyphae which spread out below ground. Now, again, fungi are a massive group that I could easily give a good couple of hour lecture on, so I'm going to miss out on most of those and just going to introduce you to some mycorrhizal fungi. These are fungi which form a symbiotic relationship with plants. That is, a relationship where they help the plant and the plant helps them. Now, this is such an important relationship that more than 90% of terrestrial plants form this relationship. It's only really the brassicas that don't. So in this image here, you can see a cross-section through the soil at the bottom of a little seedling, and you can see the root mass coming out. But all of this fuzzy stuff that you can see at the edge isn't actually root mass. These are mycorrhizal fungi. If you could look at them down a microscope, you'll see all of these thin hyphae that we saw in the previous image, although they wouldn't be glowing blue unless you stained them. So the reason why this relationship happens is plants generally get as much food as they want because they can photosynthesize. They can literally make food from the air. What plants are normally lacking is nutrients like nitrogen and phosphorus and maybe water. Now below ground it's the reverse. These fungal hyphae are very good at scavenging about through the soil because they're these thin hyphae, so they can grow very fast without too much energy. So they're great at scavenging water and nitrogen and phosphorus, but they have trouble getting hold of enough carbon, enough food. So the relationship works by the plant gives the fungus some of the carbon, and the fungus says, oh, thank you very much, and they give them nutrients in return. Now, these mycorrhizal networks, again, have been subject to research for a number of years, and they've thrown up some very surprising results over the last couple of decades. It's been shown conclusively that plants communicate with each other below ground through these mycorrhizal networks. And plants are able to pass on information such as, I'm not getting enough sunlight, or I don't have enough water, or I'm being attacked by a herbivore, perhaps a load of caterpillars or something. They can pass that information out, and other plants who are connected to the same mycorrhizal network will respond to that. Now, this can be surprising to people who haven't heard of it before. I can assure you there's certainly no pseudoscience involved. This is all peer-reviewed primary research literature. And you can see here, defense response to leaf-chewing caterpillars. So one plant that starts to get eaten will tell other plants to start doing things to upscale their defenses. At the bottom here, we have increased photosynthesis and carbon transfer. So as I say, plants can say when they're in the shade and aren't getting enough sunlight, and other plants can give them food. And it's actually been shown that plants can recognize kin. So plants will preferentially direct photosynthate, the food that they produced, to plants that are growing from seeds that they produced. So there's a whole world of below ground communication taking place. But time is limited, so on to the bacteria. So here we can see some bacteria growing on the pores of the soil. Now, as I mentioned, you get remarkable abundances of bacteria within the soil. Now, it's good to grow on the pore space because your food can get carried to you and your waste get washed away when water percolates down through the soil. But it also means any amoebae or nematodes going around can eat you. So there's not a lot you can do to protect yourself. But soil are actually quite dynamic. Aggregates break apart and reform, and so you occasionally end up with situations like this, where you have bacterial colonies growing completely encased in an aggregate. So here you can see the pore space around the outside. So if there was a protozoa or amoeba or a nematode going through here, they wouldn't be able to get in to eat these bacteria. So that's the good thing. The bad thing is that they're dependent on the rate of diffusion to bring their food in and to take their waste away. And if the food diffuses in faster and they produce too much waste before it can diffuse away, they will end up killing themselves in their own waste, which is never a nice way to go. So all of these organisms together, when they function together as we would expect them to within their particular environment, that's what gives soil a health. It's got a definition here from the Food and Agricultural Organization. Basically, it says if soils can maintain and enhance water, air and quality and promote plant and animal health as you would expect them to within their local environmental constraints, well then that is a healthy soil. So here's an example of all of these organisms interacting together. This is the nitrogen cycle. It's obviously a fairly complicated image. I'm not going to talk you through it too much, but why we need diversity within soils is all of these different steps are carried out by different species. So if we lose some of these species from soils, well, that can represent a complete loss of these biogeochemical cycles taking place within the soils. Now that's all well and good, but I quite often get a question with students like, that's all very interesting, but why should we care? Well, the reason we should all care about this is soils can be degraded. Here is a map showing a global desertification vulnerability map, with red showing the areas which are vulnerable to desertification. Desertification is a loss of soil health. It occurs when we either have too much organic matter depletion, too much contamination, or a loss of biodiversity. And it can result in massive losses in food, and basically this is what led to the Dust Bowl in the 1930s in America. So why is soil important? Why does this matter? There's surely soil everywhere that we go. Well, soil provides food, fuel, fibre and medicines, and it actually provides a surprising amount of food. Globally, more than 99% of our calorific demand is met by food that's grown in the soil. Now, locally, there can be deviations from this. If you go to a small Pacific island, they would get more than 1% of their calories from the sea. But as a global average, more than 99% of our calories are met by food grown in soil. So if we don't have soil, we don't have food and we don't have us. Now, a key thing to bear in mind is soil is a non-renewable resource. That may seem surprising because we know that soil is generated over time, but the speed that soil generates is so slow that our human timeframes is basically non-renewable. On average, in the UK's climate, it takes somewhere between 200 and 400 years to form 1 centimetre of soil. And that 1 centimetre of soil can be washed away in a heavy rainstorm if that soil has not been looked after well. Now that 200 to 400 years will give you something that if you look at it, you'll say, yeah, that looks like a soil. But actually for you to have the bio-withering, for you to have the liberation of all of the minerals and nutrients that you need within the soil to make that soil fertile, that can take 3,000 years. So we really need to look after the soil we've got. So that begs the question as to how much soil there is. Now, this image was produced by Arwin Jones, who works at the JRC, part of the European Commission, back in about 2010. And this shows in these orange colours how much soil there is. This is not the total amount of soil, this is the naturally fertile soil that feeds the world. These are basically the bread baskets of planet Earth. And the soil here accounts to about 13 to 18% of the land surface area. Now, that's not to say you can't farm areas outside of these regions, but to farm those areas, you need a lot more inputs in the form of micronutrient fertilizers, manures, and a lot more energy going into those systems in order for them to be productive. So that starts to give some perspective that perhaps soil isn't as infinite and everywhere as it's sometimes seen. But we're still interacting with the soil as very much this two-dimensional flat surface, which is somehow not very satisfactory. So a couple of years ago, I stumbled across this image where somebody was trying to counter the argument of the solution to pollution is dilution. And you've possibly also heard that humans can't be causing climate change because there's so much atmosphere, there's so much air all around us, that how can we possibly be influencing that? And that's because humans are very bad at dealing with depth. Now, obviously, with the sky is more height than depth, but you get the general idea. When you go outside, it seems that the sky stretches on pretty much forever. That belies the fact that actually the sky, the atmosphere, is a very thin layer around planet Earth. And if you were to scoop up all of the atmosphere and put it at one standard pressure, so at sea level pressure, that's how much atmosphere is keeping us all alive. Not quite as much as it's sometimes seen. I also saw another figure where somebody did the same thing with water. And again, water, there's depth to water, but actually the surface of planet Earth is relatively smooth. The deviation from the deepest part of the ocean to the top of the mountain, if you were to scale that down to the size of a snooker ball, the Earth is so smooth that it would be allowed to be used in the World Championship. So the layer of water is actually relatively thin. So these images, you can see 3 dots. The big one shows all of the water that exists on planet Earth, all of the oceans, all of the ice caps, all of the underground aquifers, everything. The second dot that you can see, the middle-sized dot, that's all of the fresh water on planet Earth in all of the aquifers, ice caps, rivers and lakes. And that third one, that little dot, that's all of the water that exists in all of the rivers, all of the lakes and all of the reservoirs. That's basically all of the drinking water that we have on planet Earth, represented there as that sphere. Now, perhaps you can see where I'm going with this, because I've often wondered just how much soil we've got and how we can visualize that. And I thought these images were very powerful, but it turned out no one had done one for soil. So I did a bit of fishing around and a bit of research, well, because that's kind of my job, to try and identify how much soil there is. Now, there's a number of estimates out there as to how much soil is used for agriculture. The lowest estimate I saw was Wikipedia, which was 1.6 billion hectares. The CIA World Fact Book came in at 3.6, but according to the FAO, who should be the authority on this, they claim 4.2 billion hectares. And I wanted to be conservative because I certainly don't want to do any scaremongering, so I thought I'd use the most conservative number there. Now that's fine for area, but what about depth? In some places, soils can be more than 100 meters deep. In London, for example, London clay goes down well over 100 meters in some places. But the vast majority of that is subsoil. So all of the nutrients and the vast majority of life exists in the topsoil, the thin layer of soil at the surface. If you were to scrape away the top 20 centimeters of soil just about anywhere in the world and try and farm the subsoil, well, you're going to have a bad time. So in terms of depth, we assumed an average depth of 20 centimeters. So with that, it's very easy to do a bit of mathematics and a bit of geometry and find out just how much volume of soil that is. Which I did, and if you plot that and you plot it on a globe as per the previous images, well, you can't really see the dot. So how much soil is that? Well, there we go. That is how much soil that is, plotted there on the UK. That's basically all of the soil, which is providing more than 99% of the calories that feed us on planet Earth. Now, I don't want to oversell this. If you're standing at the bottom of that sphere of soil, you would not be saying it's a small sphere of soil. It would be stretching up into the sky 25 kilometers, which is about twice as high as a commercial jet flies. Well, when they used to fly before COVID. So it's certainly not a small amount of soil, but what you can see is it's certainly very much finite, and it's not all that big when you consider just how important it is for humanity's civilization, for humanity's survival. Every time there's a bit of erosion, every time a bit of that soil is washed away and ends up at the bottom of a river or in the ocean, every time there's an accidental chemical spill, or every time somebody builds a new car park or a new shopping complex or housing estate on fertile soil, that sphere gets just that little bit smaller. So I'll leave you with that thought, which is possibly a bit deep for a Thursday night. So to cheer things up a little bit, I'll show you just how far some people are willing to go to investigate soil health. And with that last slide, I will thank you for your attention.
Simon, thank you very much indeed. For some reason we can't, can you hear me? Can everybody hear me?
Yes, I can hear you.
You can hear me, but I don't think anybody else could for some reason. I'm not quite sure what's going on there.
Right. Well, I hope everyone can hear me okay.
Yes, they can. Everyone can hear you fine because we get lots of questions coming in. Okay. Here we go. Hang on a second. All right. Yes.
So...
Right. I'm not sure anyone can hear me, but you can hear me, Simon. So maybe it's a sort of one-handed conversation here, but people can hear what you're saying. We've got a few questions coming in, and let me just see. Right. Let's give you the first question there. This is from Buffy Hardy. What advice would you give to farmers to improve the biodiversity of their soil? Perhaps you could repeat the question so people can hear what it is.
So the question, I'm afraid I don't remember who it was from, but the question was, how can you increase the biodiversity of your soils? And the answer to that is basically you've got to feed your soils. If you can feed your soils, the life will come. So good manure, good compost, good organic materials going into your soil is really key for supporting the biodiversity that lives there. But if you can do that, if you can get good organic materials going into your soils, well, then there's life all around and there's life moving about in the atmosphere, because soil life is so small that even if you have a very deteriorated or depleted soil community, they will rejuvenate quite quickly.
Okay, people can hear me apparently, so that's good news. And just if you hadn't already worked out, there's a Q&A button at the bottom of your screen. If you'd like to type in a question, perhaps tell us where you're from. That'd be interesting to know as well. Actually, from the same person, it was Buffy Hardy who asked the question, and that is, how can we avoid invasive species being introduced as a consequence of biocontrol?
Great question, Buffy. Apologies for forgetting your name initially. Well, we have to be very cautious with it. Many times of doing biocontrol, releasing organisms into the environment has seemed like a good idea, and there's 1,000 examples where it's turned out not to be. So no, we have to be very cautious with this. As I mentioned, the vast majority of biocontrol is done in where we use these types of organisms is done in greenhouses and places like that. And they're organisms which are naturally found in this country anyway. We just breed up the numbers. So if they were to make it out of that environment, they basically wouldn't find anything that they could eat and the numbers would die out. So we generally only use organisms which are found in the country already, or occasionally it has been done with organisms which are known to not survive the winter.
Actually, I did want to ask a question myself, Simon, because clearly we've learned a lot more, as we've heard this evening, about the nature of soil and the mechanisms that live in it. We've been dependent for so many years in terms of agriculture on chemicals and fertilizers and whatnot's going into the soil. Are we at a point where we understand enough now to maybe scale up things like the predators you were showing us to ease off a little bit on chemical fertilizers, or is that just not practical because of the pressures to produce?
No, that's absolutely possible and where we need to go. Not necessarily with the predators specifically, but by using conservation and regenerative agriculture, the idea is basically to try and keep your soils covered the entire time and use cover crops. So there's always green stuff on your soil pumping energy into the soil system, and that can increase the amount of biodiversity you have within a system. And if you have high levels of diversity, that means you'll naturally have natural predators and things within your soil communities. So the ideal thing is just to look after your soils in such a way that you have very robust and diverse communities, and that in and of itself will mean most years you can reduce the amount of fertilizer and pesticides and things that you will need.
Tom Kirby's asked the question: what area of the UK is naturally fertile? How fast can a naturally fertile soil deteriorate? I think that's something that you touched on earlier on, actually, in terms of.
Yeah, great question. Well, most of our soils are pretty fertile in the UK. If you compare them, I mean, it's all a relative term. So if you compare our soils to tropical soils, which are normally very low pH and quite sandy, all of our soils are pretty good. The most fertile soils in the UK are quite often over in the east of England, and unfortunately, a lot of our peaty soils are some of our most fertile soils. And the reason for that is that peat is organic material, and that organic material is carbon bound up with lots of nutrients. So when you manage those soils, one of the consequences of that is you burn off the carbon, which is a terrible thing in terms of climate change. But when you burn off that carbon, that liberates nutrients, which is a great thing in terms of farming. And that's why some of those soils have been farmed quite so intensively as they have.
Mark and Janet Trow with an interesting question that's very topical for anyone who knows Shrewsbury and certain parts of the county of Shropshire. How long does it take soil to recover from temporary flooding of land areas? And that happens a lot around our neck of the woods.
Yeah, well, I'm afraid the classic answer there is a scientific one, which is it depends. So it depends very much how long it's been flooded for, how deep it was flooded for, what was growing there in the first place, what you're planning on doing afterwards. There's some evidence down in the Somerset levels, for example. Some of those soils have still not recovered back to the same level of functionality as they had, and that was the serious floods they had there were, what, 3 years ago now. So some soils can take a long time. If you have a sandy soil and it's not flooded for too long and it's flooded with clean water, well, they can recover fairly quickly. Within a few weeks, you might not even notice they've been flooded.
There are some ecosystems, aren't there, in the world where flooding is part of the natural process of bringing in nutrients, isn't it?
Indeed, I mean, yeah, the Nile Delta, that doesn't exist as it is if it wasn't for flooding. And then flooding is also used for weed management in paddy fields and the like as well, of course. So yeah, it very much depends.
Charlotte Hollins has a question for us. What do you think the impact is of ploughing regularly on soil organisms? Generally, these soils lack organic matter or a soil cover. Do you think that's detrimental?
Yes, perhaps not surprisingly, if you are a soil organism, having a large piece of metal drawn through your house on an annual or multiple times a year basis generally doesn't do you or your habitat many favours. Ploughing generally often inverts the soil. That takes organisms which are adapted to live on the soil surface and puts them underneath. They don't like that very much, and it brings organisms that are adapted to living below the soil to the soil surface, and they don't like that very much either. So no, ploughing is quite detrimental. It also breaks open aggregates, so that can expose organic materials for decomposition, so that can reduce the organic matter contents of your soil over time. All the time it's doing that, that's putting more carbon in the atmosphere, so that's bad for climate change. It's also bad for your soils because that organic matter does a lot of good stuff in your soil, like holding the aggregates together and holding on to moisture and nutrients and the like.
I mean, is any work being done to find a more sensitive intervention in terms of planting?
Yeah, a lot. There's a type of drilling called direct drilling, which is part of conservation agriculture or regenerative agriculture, however you want to call it. And that doesn't involve tilling the soil at all. It's all about minimal disturbance to the soil. And I've done a bit of research on that myself at Harper. I engaged with some farmers who farm their soils like that. So yeah, there's far too few places farmed like that in the UK. In my opinion, we're at less than 5% of all arable soils are farmed like that. If you go to Brazil, more than 50% of them are, and in America as well, the numbers are much higher. So yeah, there is a shift away from the plough, certainly.
Okay. Mary Bendel has a question for you. Is there a broad rule of thumb to work out how much carbon is locked up in different soils, comparing, say, woodland with farmland or heathland? Is there some sort of equation around that? Or?
No, people have tried, but again, the classic answer is it depends. Unfortunately, a forested area on a sandy soil will have less carbon than a forested area on a clay soil, but both of those will probably have more carbon than, well, certainly have more carbon than an arable system, quite probably have more carbon than a grassland system, but it really depends on the legacy of how things have been going on, and things such as relief, whether they're north facing, south facing. That affects the temperature. All of these things have a large effect. So people have tried to come up with general rules of thumbs, but there's normally as many or more exceptions to that rule as the rule works for.
Chris Brooks asks, how is modern farming with pesticides and mass production and the lack of rotation of crops and mixed farming affecting the soil?
It's not done it any favours. Most of our soils around the UK are quite depleted in organic matter, so that means they don't function as well as they could previously. They're more susceptible to environmental stress, and yeah, they erode more readily. So no, all of these things have been less than ideal. The disconnect between livestock and not returning manures and things back to the soils as we used to has really been quite detrimental over time. And that does mean that soils have a potential for soil carbon sequestration. We have, most of our soils are depleted in carbon. That means we can store more carbon in them before they can become saturated. So there is some potential to use them as a tool to try and mitigate climate change. Although I hasten to add that the potential that they have is quite often overstated in the popular media.
Tim Dawes has got a question that scales things right back to the allotment or indeed into our own back gardens. If we're growing veg, is no dig best for soil health?
Again, the answer is it depends on the soil that you're working with and the plants that you're planting. Personally, yes, minimal disturbance to soils is generally a good thing. The biology that's in the soils will appreciate it, unless you have massive issues of compaction. For example, if you're working in an allotment and the person that worked there previously has beaten your soil to pieces, then tilling it and mixing it up and getting some manures into it will probably do more benefits over the long term than disturbing the soil would do in the short term.
Tom Kirby has a question asking your opinion on the concept of high pressure water drills.
As in seed drills?
I'm assuming that possibly is the case.
I've not come across them, if I'm honest.
Okay.
So, yeah, so I couldn't comment on those. Yeah, don't know.
That's fine. Just going back to Charlotte's question earlier on about the ploughing, and I should have mentioned, I'm pretty sure that Charlotte's a relative of Arthur Hollins, who was famous for his revolutionary techniques, which unfortunately haven't been taken up by too many people. But she's basically saying that they believed for a long time at Fordall Farm, and is this being passed on to farming students at Harper Adams?
So conservation agriculture and
the whole ploughing, the sort of reduction in ploughing, or indeed not ploughing.
Yeah, no, absolutely. I take students over to the field site that we have set up, whereby we run an experiment doing comparison of till versus no till. We also have a long-term traffic and tillage experiment set up on Harper's campus, which has been going for 9 years, where we've been looking at tillage versus no tillage and controlled traffic versus not controlled traffic. So yeah, no, we very much try to keep Harper students up with the times, up with modern research in soil management.
So that's an interesting point, that because Simon, you often hear that the younger generation are more switched on to this kind of thing. Do you get any sense at all that young people are coming through the system, through the system at Harper Adams, for instance, with perhaps a broader perspective, a wider horizon, a different view of these things?
No, absolutely. The majority of younger students are starting to become more switched on. There's still some resistance there. It's probably fair to say the farming community is reasonably conservative, and there are quite a few people who do things just because that's how it's always been done. And there are some difficulties that we need to push back against in terms of farmers' perception. For example, I've heard from many farmers that springtails are pests and they try to wipe them out. And as I say, I've never come across a single species that is known to be a pest. Another classic one is the myth that if you cut an earthworm in half, you get 2 earthworms. So I've heard farmers say that ploughing and power harrowing your soil is good for worms because you end up with more earthworms. And just in case anyone is unsure as to whether that is a myth, I can put that to rest for now for sure. If you cut an earthworm in 2, the best thing you will get is a dead bit of worm and a worm with a sore bum, but most likely you'll end up with 2 dead bits of worm.
Dan Wrench, hi Dan, good evening, has asked, can soil health be improved with inoculation with soil fauna from healthy soils?
There has been a bit of work done on this. People have been trying to introduce earthworms, for example, which are known to have been found in that area, but which have become depleted. And there is some evidence that they stick around and increase soil health over time. The difficulty there is, well, if your soils are depleted, you're normally depleted across farm scales. So finding enough biology that you can re-inoculate it is difficult. And generally, you're much better off just starting to look after those soils, and they will regenerate quite quickly because there's a lot of soil life moving around. A lot of these calendula that can run across the surface, they migrate relatively quickly. So yeah, soil life will come back as long as you start looking after your soils and feeding them.
And a supplementary question from Dan, what impact does biochar, does it have on soil fauna?
Yeah, but there's not an awful lot of research on biochar. So for those of you that haven't come across biochar, there's the idea of taking a charcoal-like substance and putting it in soils in order to lock up carbon in those soils. And there's actually evidence from the Amazon basin, a type of soil called terra preta. Lots of people who are proponents of biochar think that we can try and create these terra preta-like soils in the UK. I've done quite a lot of research with biochar. Most of the evidence suggests in UK soils, it doesn't actually do an awful lot. Most of our soils are near their yield maximums anyway, and quite rich with life. And biochar sticks around in soil for a long time because it's not decomposed. It's technically, we call it recalcitrant. So that's why it's used for carbon sequestration. It just stays in soil for a long time. But the fact that it's not consumed means it doesn't really interact with the biology very much. There are exceptions depending on what you make it from, but as a general rule, it doesn't interact with mesofauna and macrofauna very much.
And a question about carbon. Can we, this is from Buffy Hardy, can we trap carbon in the atmosphere and return it to the soil? Is there a mechanism by which we could possibly do that at some point?
Well, that's what plants do, really.
Yeah, exactly.
So yeah, that's the most efficient way of doing that. Cover crops are used quite a lot within farming these days, or being used more. That's the idea of basically growing a crop, not a cash crop, not something you're going to harvest. You just grow a crop to feed the soil. So it's grown, and then either it's sprayed off with glyphosate, which some people don't like, or it's driven over with a crimper roller to try and crush the plants up, and then they're left in the field to decompose. As they decompose, most of the carbon will go back to the atmosphere, but some of it ends up protected within the soil. That's the most efficient way of doing it. There are other people that try to do carbon capture with storage and things like that. I'm not a big fan of that. I prefer trying to reduce the carbon going into the atmosphere in the first place, because trying to capture it back from the air is very energy intensive. And actually, two of the major companies that are trying to do that, they capture the carbon back from the air in the form of hydrocarbons, which they then plan on using as a fuel, which is just genius.
And round we go.
Yeah, round and round we go, ignoring the fact that it takes massive amounts of energy to get that carbon out of the atmosphere in the first place. So it's a massive, yeah, it doesn't make sense at all.
Marvellous. Well, thanks everybody for your questions this evening, and thank you indeed, Simon, for a fascinating talk. It really is. I have to say that the idea of talking plants might be a bit problematic for vegans with a conscience, but let's not go there. Well, it only remains for me to thank our speakers, Dr. Simon Jeffery, for being here this evening and giving us such an entertaining talk, and to you indeed, wherever you are, and joining us this evening, and to invite you to join me tomorrow at 12 noon on Darwin Day to drink a toast to Charlie. You can find the details on that event, and indeed all the forthcoming events in the Darwin Festival in Shrewsbury at darwin.originalshrewsbury.co.uk. Simon, thank you very much indeed, and thanks everybody else. Good evening.
Yeah, thanks everyone for your time.